Welcome everyone to our renal grant rounds. We will start with a very important announcement. So I want you, to remind you that the third annual symposium on the kidney urology and hematology research is coming. Please mark your calendars for May 22nd. There's going to be a keynote presentation from Dr. Nancy Speck and a great poster session. So please, register, submit your poster. The deadline has been extended to May 8th. It's a great event for networking and having, a fun science day. So, with that announcement, I will switch to introduce our very special guest speaker for our renal grant rounds. So it's really my honor to introduce Dr. Alexander, or Sasa, Staruschenko, who is a professor and director of the Hypertension and Kidney Research Center at the University of South Florida. The research of his laboratory is really focused on understanding mechanisms that control the activity of ion channels and electrolyte homeostasis, regarding control of blood pressure and kidney disease as well. And, we all know how hard is the funding environment. He has been able to have continuous funding through the NIH, VA, American Heart, and this is because of really, hard and exciting work that has led to more than 175 papers in leading nephrology and hypertension journals. So Dr. Staruschenko, along this amazing work, he has mentored several students, postdocs, junior faculty, and really he has been dedicated in this field, and that has led to national, international recognition as well, with multiple invited talks all over the world, and distinctions, awards by the American Society of Nephrology, the APS. He has been a deputy editor for the American Journal of Physiology, past chair of the American Heart, Kidney, and Cardiovascular Disease Council, a standing member and in multiple NIH study sections. And, really there are a lot of things to say, but, I found out yesterday that his journey started from St. Petersburg in Russia, and apparently now he's very close. He's in Tampa, and he can see the St. Petersburg. Is that right? Yeah. In Tampa, and he has been able, like it's what I learned is that we know that life is very short. He has been able to do a safari trip while he managed to transfer his lab and continue his papers and his research and all this during the COVID pandemic. So there is always, opportunity to do things. So I will stop here. Thank you, Sasa.
Thank you very much for a very nice introduction and for this opportunity to present at Northwestern University. And I was actually your neighbor and for almost 15 years, and I spent 14 and a half years at Medical College of Wisconsin in Milwaukee in Wisconsin, but never had a chance to visit Northwestern University. And probably you need to move from St. Petersburg, Russia to St. Petersburg, Florida to be invited to Northwestern. And anyway, and I'm happy to be here, and I'm happy to present some of our recent data. And also I'd like to start with a statement that my research is very unfocused, and I mean that I'm unfocused by nature, and I just do whatever is scientifically interesting, and sometimes it works, sometimes it doesn't, and I consider myself as an ion channel guy and who focused on renal ion channels, but currently I have 0 ion channel finding, and but I still do ion channels physiology, and also let's say that in last fall, and I was asked to give a talk at ASN about lysin metabolism. And next week, and I gave a talk at AHA on opioid signaling in the kidney. And months before, and I spoke and at AHA hypertension scientific session about SGLT2 inhibitors and salt-sensitive hypertension. And week before, and I spoke on German Physiological Society about potassium ion channels in blood pressure control. And for me, it was hard to pick and what I actually have to present today. And but I decided to talk and about some potential mechanism controlling and playing a role in diabetic nephropathy. And also, I want to apologize in advance, and that this presentation will be completely unfocused, and I mean that it will be some project where I will not present enough details, and because it was my desire to show you a little bit of everything, and as a result, and okay, we have what we have, and. Anyway, and here's my only background slide, and that diabetes and is a huge problem, and nationwide and worldwide, and it's almost 35% of US adults and have pre-diabetes and develop diabetes and at some stage of disease. And also, it's a serious complication which results in the end-stage kidney disease. And I want to tell you that there are a lot of people who actually focused on diabetic research, but there are not enough people who actually do diabetic nephropathy or diabetic kidney disease. And I served for a number of years in the American Diabetes Association study section. And they have a completely, I would say, stupid and way how to organize it. They send all reviewers and abstracts and titles of all grants submitted and to that cycle, which is about 600 abstract. And even if you spend 30 seconds per each, and it takes some time just to go through all of them. But what I want to tell you is that only about 20 or 25 and out of 600 have anything to do with kidney. And despite that all these diabetic patients and have severe and kidney issues, and in many cases, they actually die because of end-stage kidney disease or go to dialysis. Anyway, and I'll stop here with my background, and here is my outline of my presentation. And first, I will try to advertise you a little bit T2D rat model, which we used in our research. And it's not the only model, and which we try to work with DBDB mice, and we used STZ injected hypertensive rats and a few other models. But we ended up with this model as a very good animal model to do diabetic nephropathy studies. And here is an initial paper and which characterized this model. And it was published in 2004 in journal Diabetes and by a group from Medical College of Wisconsin, where I used to be. And basically what they have done, and they took GK Gotakakizu model, and which is a well-known diabetic type 2 diabetic model, and we backcrossed it with FHH rats, full-hooded hypertensive rats. And basically GK rats, they have type 2 diabetes, but they don't have kidney injury. And since they begrow them with FHA rats, and they had 98% of genetic background from GK rats, but got some additional genes and from FHA rats, and those rats and actually started to develop and diabetic nephropathy. And at the same time, and basically Howard Jacob, Rich Roman, and they filed a patent, and I think they made a mistake, and because they tried and commercialized in this model because at that time and even now, and there are not enough good models to study diabetic kidney disease, and they try to tell in the scientific community, okay, here is a great model, and try to get a patent and actually sell it. But you know, all of us and academic scientists don't like to pay for anything, and they want free stuff. And as a result, scientific community say, oh, you know what, and this particular and populations for glomerular damage is not exactly the same as you see in human, and or this particular stuff started a little bit later than needed, and basically it's, I would say it was not well accepted. And but key point is actually here that this patent was filed and in July 2003, and it's expired in November of last year. And I spoke with Rich Roman, and he told me that he is going to deposit this RED model to RED depository where everyone will be able to get it just to pay for shipment or whatever. And if not, and feel free to approach me, and I'm happy to provide you any samples, and because we have everything from different age, sexes, and all different organs, and or we can ship you live animals as well. Anyway, and what's about this model? And basically, here is some slides where I want to show you and some comparison from original studies published 20 years ago, and also some of our studies where we also compared and these rats and with other diabetic or non-diabetic models. And here and from a slide from original study where they compared it to Brown Norway and non-diabetic, non-hypertensive model, or to GK or von Hooded hypertensive model. And you see that these animals and start to develop and proteinuria and a certain age and about 12 weeks old. And also they have and different glomerular damages and at least partially corresponding to what you observe in human. And also we compared and we've done different analysis. We checked the glucose level and total body weight, and also we analyzed protein cost and diuresis in these animals. We checked and how they what kind of injuries they have, and for this analysis we compared T to the N to GK diabetic rates and also to V-star or WKY and non-diabetic rates, and we used V-star as a non-diabetic rates as a control because GK rates were developed from V-star rates and down the road. And here and we also will quite surprised and to see a huge difference in sexual dimorphism in these animals. And basically, here and you can see some data with glucose tolerance test, with blood glucose and serum and serum albumin level and albuminuria and male, female, and younger and older age. And when I will use younger and older for this particular presentation, younger means about 12 weeks old, and older means and more than 40, 48 weeks old. And also megalin staining, where you see that megalin and in diseased model and was expressed not only in proximal tubule, but actually was transferred to Bowman capsule. And what I want to show you here, actually, that male and T2DN rats, yes.
Are you saying there are male differences in the slide between male and female, and then yet which one is more
Okay, basically here is a female staining and on the bottom and a male on the top. And what I wanted to show here, which it wasn't what we expected to see, but basically male T2D rats, especially at older age, and they develop a severe diabetic kidney disease. Female, and they also develop a kidney injury, but much lesser in extent here. And when these animals develop this kidney injury, what we see here, and it's nice and staining for megalin and proximal tubule, but in addition to that, we see this nice and move and staining of megalin and Bowman capsule. And I would say that, and a similar observation were observed in some human biopsy samples, and were very well described by Agnes Fogger, and when she reported and megalin transformation from proximal tubule to Bowman capsule parietal cells, and similar we observed in this male T2D rats.
Can you say that females have less megalin expression in the lower panel? Or is it
I don't want to say that they have less megalin expression. I want to say that male compared to female, and in addition to expression in proximal tubule, also have an expression of megalin in the Bowman capsule.
The proximal tubule is more targeting that is well known to be the pathway for albumin uptake.
Yeah, and for.
They don't go in the
Mm-hmm. Yeah, and for protein, for proximal tubule, and here, and here, it's a good example, and it's Western blots and summary on the right, and for KIM1, which is a marker and for proximal tubule damage. And you see that young male and have a little bit more than female, and older male have a dramatic increase in the KIM1 staining, and showing that proximal tubule were severely damaged in these animals. And also we saw a difference in nephrin. Here it's a nephrin in kidney tissue and not in urine, and basically decline in nephrin expression showing less and function of glomerular cells. Old means and more than 40, 48 weeks old. And I mean that it's old, but not old enough, probably my age. And I mean that, but also for T2D rats, at least for male, and we've done some analysis for 60 weeks old, and their kidney is a mess. And I mean that even at this age, and we have all these differences. And anyway, and basically, first part of my presentation is to try to convince you that it's a good model to study diabetic kidney disease, and also this model has changes in some sex difference, and unfortunately, NIH might not appreciate this because male actually have more damage than female. And it's a little bit easier to sell your grant if you tell NIH that female have more issues and I want to study female. And but we usually say that, okay, and actually for human, and it's quite often, and basically observations that male and diabetic kidney damage is more prevalent and than female, which is still and a little bit controversial, but at least it's a main and assumption. Anyway, and next, and I'm going to talk a little bit about cGAS-STING pathway, and which is a quite important pathway, which was reported a number of years ago, and we came to this pathway when we initially performed RNA-seq analysis and of both male and female, and at younger and older age, and basically, what I want to tell you is that, and when we performed IPA analysis of this RNA-seq data, and we found that many genes and almost everything in this pathway was upregulated in male aged and compared to female, and also even younger male and compared to female, and I'll show you some of this data. And when we basically performed this RNA-seq analysis, we decided to explore this pathway a little bit. And we took human kidneys and healthy human kidney and type 2 diabetic human kidney, and as well as non-diabetic Wistar and T2DN rats, and we analyzed for STING, and one of key proteins in this pathway. And we found that T2DN rats and have much high expression, and also human with type 2 diabetes, and also have high expression of STING. And also we performed Western blot analysis and for different proteins in this pathway, cGAS-STING and TBK1 and IRF3 and T2DN rats compared to non-diabetic Wistar rats. And most of these proteins were highly upregulated in T2DN, some and didn't reach a significance, and but still and had some trends and in T2DN rats compared to Wistar rats. And here it's also, and it's younger animals. And I mean that if we perform the same analysis in older animals, and I'm pretty sure that we'll have a much higher, bigger difference. And also we analyze the same proteins and in young female, oops. Oh, no. And okay, this way. Okay, in young female, young male, and old female, and old male, and pretty much all of these proteins were much had a much higher expression in the old male compared to female, and also in old compared to young, and which gave us an opportunity to conclude that this pathway might at least potentially and contribute to progression of diabetic nephropathy, and definitely play a role, and might be potential targets and for diabetic nephropathy. And since cGAS-STING and pathway is involved in the inflammation, we performed a flow cytometry analysis, and again in the same 4 groups of animals, and we analyzed different cell types, and you see that it's data for T lymphocyte and B cells and T cells, macrophages, and pretty much in all groups. And we observed that, and in the young male, and it was elevation compared to young female, and in older male, and it was even further elevation compared to young male. And basically when we observe this kidney injury, and we have a severe increase and in inflammation pathway, which at least partially might be mediated by cGAS-STING pathway. Here is in our brief summary and of what happened in male and female, and how this pathway and might be activated. And of course, and it's a lot of unclear stuff which should be addressed, and some are just hypotheses, and whether it's a mitochondria involved, whether some other organelles involved, and how all this pathway and inflammation actually play a role. And but at least, and I hope we can conclude that cGAS-STING pathway and inflammation play a major role, and in the progression of diabetic kidney disease, and also much higher elevated in older and with age and in male compared to female as well. And with that, and I'll move to another and part of this project, which is also focused on diabetic nephropathy, which is also using and T2DN rats, and but it's about purinergic signaling. And how we came to this idea, basically, it was shown and in a number of pathophysiological states, and including hypertension, glomerular nephropathy, and polycystic kidney disease, inflammation, that changes in purinergic signaling receptors might contribute and to development of different pathologies. And our hypothesis was that remodeling of purinergic signaling in podocytes might play a crucial role in diabetic nephropathy. And again, we used T2DN rats, at least partially, to address this. We've done additional study with few other models and also cell culture. And here I'll show you what we've done with T2DN rats. And for those who are not aware of purinergic receptors, there are two types of purinergic receptors. There are P2X and inotropic and purinergic receptor, and P2Y receptors, and which are metabotropic receptor, typical G protein couple receptor activated, and basically. When ATP or any other stimuli and activate and metabotropic receptor, it's activate this entire G protein couple receptor mediated cascade and result in different effects. And with P2X receptors, and it's almost like activation of ion channels. And basically ATP activate P2X receptor, and it's and P2X receptor, and able to conduct calcium and some other ions, mostly calcium, and into the cell. And for this project, we mostly focused on podocytes. And here I show you some healthy podocytes, and there are some hypertrophy food process, and which are mediated by elevated intracellular calcium, and which result in podocyte death. And interest to this area, and specifically for calcium and podocytes, started back to 2005, and when two independent group, and one at Duke and one at Harvard, and identified and human mutation in patient with FSGS and in TRPC6 channel, and which cause focal segmental glomerulosclerosis. And we were interested to exploring this topic. And to do this, and we actually were able to develop a new approach how to analyze and calcium flux in podocytes. And basically, we isolated freshly isolated decapsulated glomeruli, and during different sieving, and which is shown here, basically take any type of rat, mice, and go through different sieves. And at the end, we have this freshly isolated decapsulated glomeruli, which we applied and found a specific condition where we can do calcium imaging and electrophysiological analysis, where we performed ion channel analysis in the body of those podocytes and those glomeruli. And I would say that our lab was the first and who actually established this approach. And since then, and many labs, not only in academia, but also in industry, and apply this for different imaging and electrophysiological analysis. And here is actually a funny picture which I want to show you. And in the middle, it's me, and on the left and right, it's Roger Chen and Erwin Nair. And Chen got a Nobel Prize and for development of GFP fluorescent marker, and Erwin Nair and got Nobel Prize and for development of patch clamp. And basically, I make a fun of this picture that electrophysiology and imaging and are two my favorite approaches which I use in our research. And And actually, when I took this picture, and it was also Tom Zuthoff and another Nobel laureate, and on that picture, oh no, and standing, and I asked Tom, would you please stay a little bit away, and because I want to take a picture with those two guys, not with you. I'm not doing neuroscience, and I'm not interested in synaptins and all other stuff, and which was probably a little bit rude. And anyway, and here is our data with initial analysis of basal calcium in podocytes of freshly isolated glomeruli. What you see here, and that in citoyen rats, and even at a younger age, and 12 weeks old, the intracellular calcium was already elevated. And I can tell you that between 250 nanomole and 150 nanomole, and it's a big difference for cell. And basically, even at a younger age, and these rats and already have some kind of elevated calcium, which we think is important and for glomerular and kidney function in general. And here you see a video, and when we just apply ATP to freshly isolated glomeruli, and you should, did I miss it? And or it was a spike in green, and basically when we apply ATP, we immediately see and we immediately see in the elevation of intracellular calcium. Okay, application. Okay, kind of. And I don't know why it's because here is, and you can see, and before and after ATP, and we analyzed effect of ATP, and again in Wistar and citoyen rats, and we see a nice elevation, and but in citoyen rats, and it was kind of prolonged response. And also we performed a dose response, and in both and younger animals and older animals, and what I can show you is that what's the difference and between GK and the Wistar, and it was some elevation and shift of this dose response curve and from Wistar to GK, but in citoyen rats, and actually even low concentration of ATP resulted in the very high elevation of calcium in glomeruli. And we saw that, and it might play a critical role. And we published some time ago in the manuscript, and where we identified a metabotropic P2Y1 receptor as the main receptor responsible for ATP flux and mediated calcium flux in podocytes. And here we checked and P2Y1 receptor expression, as well as expression of several other receptors. And again, in Wistar, GK, and citoyen rats. And we found that expression of P2Y1 was significantly decreased in both diabetic groups. In contrast, we see a very nice increase in both P2X4 and P2X7 receptors. It's the reason why I'm telling you that basically in a diabetic state, and it's a change of purinergic receptor profile and from metabotropic to inotropic, and those P2X4 and P2X7 receptors might conduct this calcium. And what we see here, and basically we apply ATP, we see a nice response, and we apply different drugs and with attempt to block it. When we blocked P2X4 receptor, P2X7 receptor, and we had almost half and inhibition of this ATP calcium flux in response to ATP. And when we blocked P2X4, we also have a nice decrease. But when we applied these drugs combined, we almost completely blocked elevation of calcium in response to ATP. And here's our summary of second part. And basically, under normal condition, ATP activates this P2Y receptor, which starts this intracellular signaling cascades, which result in some elevation of intracellular calcium. in diabetic nephropathy, and basically P2Y receptor is still there, and they still play some role. But in addition, we have P2X receptor and P2X4 and P2X7, which add this elevated calcium flux and in this model. Okay, here is our conclusion that under diabetic condition, we have increase in of ATP, which we also show it. We measure it in the ATP level in diabetic animals, and it's elevated, and it's a change in profile. And we also saw an increase in expression of TRPC6 and channels, which all result in elevated intracellular calcium and potential podocyte damage. And with that, and I have a few minutes and to briefly touch and another potential mechanism, and it's a mechanism mediated by protease-activated receptor family, and basically it's a family of receptors, and which are also G protein coupled receptors, and they play important role in blood and coagulation and this type, and but also they are expressed in the kidney and potentially play some role. And initially we checked and if those receptor and specifically PAR1 and indeed expressed in podocytes, and we measured it in isolated glomeruli, and we co-stained it with nephrin, and I believe, yeah, and we showed that yes, and it's colocalized with nephrin, it's expressed in podocytes and of glomeruli. And also it's elevated and PAR1 expression in human diabetic glomeruli. And what we actually have done, and we took a culture model and a human podocyte cell line, and we applied and some PAR1 agonist, and here specifically we applied TFLR. And we saw a nice increase. And when we gave in the cell culture human podocytes and high glucose, we saw elevation of calcium and much bigger, much higher response to PAR1 activation, and which is partially summarized here. When we blocked PAR1 receptor with RWG, and we were able to eliminate this effect and block PAR1 mediated elevation of calcium in podocytes. We also gave thrombin and endogenous and compound, and which might activate and PAR1 receptors, and we also show it in here in the isolated glomeruli. We when we do cell culture, we always try to confirm it with freshly isolated cells or freshly isolated tissue. And here you see the thrombin also resulted in very nice and much bigger increase in T2DN rats compared to Vista rats. And also we measured PAR1 expression, and it was elevated and in T2DN old compared to T2DN young animals, and compared to non-diabetic or diabetic control animals. And we also performed ion channel analysis of TRPC channels, and in response to thrombin, basically when we isolate glomeruli, perform cell attach recording, we apply thrombin and see activation of ion channels. And here you see that TRPC6 channels are much higher expressed in T2DN rats compared to GK and V-star, and basically activation of PAR1 receptor result in activation of TRPC channels. And to confirm and selectivity of the channel, we used and blocker of TRPC6 channels, and which almost completely inhibited and PAR1 mediated activation of this channel. And similar, and here is our idea and about potential involvement of PAR1 receptor, and again, and it does activate TRPC channels, and all these proteins are elevated and on hyperglycemic condition and result in all this in elevated calcium in podocytes. But again, podocytes are not the only cell type in glomerule, and we were interested in if we can see in the same type of effect in mesangial cells. And here you see application of PAR1 in the human in mesangial cell culture. And first, you can see that it's a nice elevation of intracellular calcium in terms of activation of PAR1 signaling. But also, it's a big difference between podocytes and mesangial cells. Because in podocytes, we apply and PAR1 agonist, and we see a nice peak, and sometimes it has some delayed curve and go to plateau, and a little bit later some delays, but it's just one peak. And here, it's slightly different because we see both increase. Initial peak, and next, this kind of oscillation, and which represents some kind of second wave. And we were curious what actually what does it mean. And here in the shown summary of first wave and second wave, and under two conditions, when we use 2 millimole calcium and extracellular solution or 0 calcium. And the second wave was observed only when we use 2 millimole calcium. And basically, when it's 0 millimole and there's no calcium outside of the cell and nothing what can go inside. When it's 2 millimole and it's show us that, okay, and it's this calcium goes through some kind of plasma membrane channels. And we performed electrophysiological analysis when we applied TFLR to activate PAR1 in mesangial cells. And we used different inhibitors here, and it's only one set of inhibitors which we used, but we applied multiple to test all possible and ion channels which were previously reported in mesangial cells. And what we showed that when we inhibit two types of channels, and STIM1 or I1 and TRPC3, and we were able to completely inhibit and PAR1 mediated pathway. And here is actually a summary of our findings in mesangial cells. And again, we are able to activate PAR1 receptors and through some endogenous compounds, and which result in activation of this G-protein couple signaling and activate TRPC3 channels. And in contrast to TRPC6 and in podocytes, and again, we tried different inhibitor of TRPC6, and they didn't block this effect. And also STIM1 or I1 and channels are involved in these effects. And finally, we were curious, and what happened with this signaling and in vivo. And for this, we tried to knock out PAR1 in T2D rat, and the main limitation of rat models, and it's genetic is a little bit behind of mouse models, and basically with mouse you can do whatever you want, and except for probably mesangial cell specific because there are no promoters. And with rats, and we can do knockouts, we can do knock-ins, and with knock-in of different mutations or all kind of stuff, but not cell specific yet. And it's active work on this, and I hope in near future we will be able to do same type of experiments as with mice. But anyway, here we knocked out PAR1 in T2D rats, and knockout was not viable, and they died. And it's the reason why we used heterozygotes and for PAR1. And what we showed here is that albuminuria and was not completely down, but at least partially was low in heterozygous animals. And also, and glomerular injury was a little bit less and in heterozygous animal. And as well, and as effect to activation of PAR1 signaling was low in heterozygous animals. And basically, which all show us that yes, and this pathway and crucial and for diabetic nephropathy progression. And with this, and here is a summary of my last, and that under diabetic condition, we see increased and serine proteases and PAR1 expression, which mediate calcium dynamics, and they mediate this dynamic differently in podocytes and mesangial cells. And I hope, and we are able to identify specific channels involved in this mechanism in glomerular. And here's my final summary. First, and with all this example, and I hope I was able to show you, maybe not convince you, but at least show some examples and of using T2DNRET and as a model of diabetic nephropathy. And importantly, this model is not non-obese, and because not everyone who has diabetes and has a high BMI, and there are some people, non-obese people, and it's also should be studied. And also these rats and have sexual dimorphism and might be helpful and to study this sex difference between sex differences in diabetic nephropathy. And also, I showed you 3 examples and of different pathways and which are modulated and under diabetic nephropathy condition. What I want to tell you is that we all try to focus on this particular gene, on this particular protein, and on this pathway, and but in reality, and it's more complex, and basically, and I showed you 3 examples, but of course it's not only examples, and of course other pathways and other mechanisms and are involved and mediated and play major role in the progression of diabetic nephropathy. And I'll stop here, and we'll thank and all people who actually did this experiment, and current members of my lab, and I have a group of very talented people, and also a number of former trainees and who were involved in this or other project in the lab, and right now in different parts of the country or outside, and I'll be happy to answer your questions.